The scientific method, evidence-based medicine and visual processing. The scientific method is one of the most successful intellectual tools ever developed. It allows us to separate observation from assumption. We formulate a question. We develop a hypothesis. We make a prediction. We test it. We measure the result. We analyze the evidence. And, critically, we attempt to falsify the hypothesis. Where the variables can be defined and controlled, this works extraordinarily well. It has given us much of our understanding of optics, retinal physiology, visual acuity, contrast sensitivity, eye movements and many other aspects of vision. But the scientific method does not become wrong when we move into visual processing. The problem is that the thing we are trying to study becomes much more difficult to define and measure. That distinction is fundamental. In many visual experiments, we try to isolate one variable. Change contrast while keeping everything else constant. Change spatial frequency. Change luminance. Change color. This is good experimental methodology. But visual processing is not necessarily organized into independent variables. Optical quality may interact with luminance. Luminance may interact with contrast. Contrast may interact with spatial frequency. Color may interact with adaptation. Stimulus duration may interact with temporal processing. Fixation may interact with attention. And attention may alter which information is selected. These effects cannot necessarily be added together. The combination can produce an effect that is different from the effect of any individual variable. This is an interaction. And as the number of interacting variables increases, the number of possible combinations increases rapidly. Therefore, an experiment can produce scientifically valid results about individual variables while still failing to describe what happens when those variables occur together in real visual experience. Reduction, the strength and the weakness. This creates an important paradox. Scientific experiments need reduction. We simplify the stimulus. We control illumination. We restrict the task. We control viewing distance. We ask for a specific response. This gives us experimental control. But there is a trade-off. The more variables we remove, the less of the real visual situation remains. That does not make the experiment bad. It defines what the experiment can tell us. A laboratory experiment may establish what happens when one particular stimulus characteristic changes under controlled conditions. It does not automatically establish what happens when several characteristics change together in everyday vision. This is especially important when symptoms occur only under particular combinations of conditions. Then we reach perception and qualia. There is an even more fundamental problem. What if what we are trying to understand is what it is like to see something? A person may report that text appears blurred. Another may report that it moves. Another may experience it as unstable, distorted, doubled, fragmented or unusually bright. We can record the person's report. We can change the stimulus. We can investigate whether the response is reproducible. We can measure behavioral and physiological correlates. But we cannot directly measure the person's subjective experience in the same way that we measure luminance or wavelength. The scientist observes the stimulus. The scientist observes the response. The person experiences the perception. Those are three different things. They cannot simply be treated as equivalent. This becomes even more difficult with phenomena such as unusual visual associations, synesthetic experiences, spatial distortions or altered perception. These experiences are not therefore unscientific. But the evidence is necessarily more indirect. A person's report is data, but it is a different type of data from an instrument reading. Evidence-based medicine is extremely important, but it is sometimes misunderstood. It does not mean simply following whatever has the largest study or the most impressive statistical result. Its purpose is to integrate the best available research evidence with clinical expertise and the circumstances of the individual patient. And this distinction becomes critical in visual processing. The strength of evidence depends partly upon whether the research actually addresses the question we are asking. Suppose there is strong evidence that a particular intervention improves visual acuity. That tells us something about acuity. It does not automatically tell us what to do with someone whose principal problem is visual instability, or apparent movement of text, or distortion, or unusual responses to color or contrast, or difficulty sustaining visual attention, or severe difficulty with everyday visual tasks despite good conventional acuity. The evidence for one measurement cannot automatically be transferred to another phenomenon. This creates a fundamental danger. There is insufficient evidence for this phenomenon can easily become confused with this phenomenon does not exist. Those are completely different conclusions. Evidence gaps are not proof of absence. The evidential base itself is shaped by what researchers choose to measure. If research predominantly measures acuity, refractive error, contrast sensitivity and other conventional endpoints, then those measurements will naturally dominate the evidence base. But phenomena that are difficult to standardize are much harder to investigate. How do we quantify visual movement? How do we measure distortion? How do we quantify a perceptual experience? How do we measure the interaction between color, contrast, spatial frequency, temporal frequency, attention and individual processing? These are difficult scientific questions. But difficult to measure does not mean impossible to investigate. It means that better methods may be required. We can manipulate the stimulus. We can change illumination, change contrast, color, spatial arrangement, duration, temporal characteristics. Then we can ask whether the person's performance or experience changes systematically. That converts a subjective complaint into something that can potentially be investigated experimentally. Why this matters in optometry? This brings us to optometry. The optical and refractive basis of optometry is not fundamentally flawed. It has a strong scientific foundation. Refraction can be measured. Optical errors can be characterized. Lenses can alter the optical image. Visual acuity can change as optical quality changes. These relationships are experimentally accessible. The difficulty begins when the same conceptual framework is extended to symptoms that are not primarily refractive. A patient may report visual instability. Words may appear to move. Letters may reverse or invert. The visual scene may appear distorted. There may be unusual sensitivity to particular visual stimuli. There may be difficulty sustaining visual attention. There may be a mismatch between excellent acuity and severe difficulty in everyday visual tasks. These symptoms cannot simply be assumed to be refractive because they occurred during visual activity. The fact that a symptom is visual does not establish that its cause is optical. A refractive correction changes the optical image reaching the retina. It does not establish what happens to that information afterwards. The visual system continues through retinal processing, neural transmission, cortical processing, integration, attention and perception. Therefore, when an appropriate optical correction does not explain the problem, the scientific question should become: what component of the visual process has not yet been investigated? The real scientific issue. This brings us back to both the scientific method and evidence-based medicine. Every test has a domain of validity. A test designed to measure refractive error should not automatically be treated as a test of neural processing. A test of acuity should not automatically be treated as a test of visual perception. A measure of eye movement should not automatically be treated as a complete assessment of visual attention. And a behavioral response should not automatically be treated as a direct measurement of a neural mechanism. The problem is not necessarily that the measurement is unreliable. The problem can be over-interpretation. A valid measurement can be used to support an invalid conclusion. So we need to separate three things. what the person experiences, what we can objectively measure, and what we think explains the relationship between them. Those are not the same thing. The scientific method remains our best tool. Evidence-based medicine remains essential. But both require us to recognize the limits of the evidence. In complex visual processing, multiple variables interact, effects may depend upon context, and some of the phenomena being investigated are subjective. Therefore, the evidential chain from stimulus processing perception mechanism can be exceptionally difficult to establish. The answer is not to abandon science. It is to develop better questions, better stimuli, better measurements, and better evidence. Because the absence of an adequate measurement is not evidence that the phenomenon is absent. And an inadequate model should never be mistaken for an adequate explanation.